Hi, I'm Jean, and I'm here to help you understand what gene therapy is.
Good day, everyone. For today's lesson, our group would like to discuss about the gene therapy. Stated clearly represents what exactly is a gene. Each of our cells contains 46 strands of DNA. A single strand is made up of millions of particles called nucleotides. And these nucleotides come in four different types which scientists have labeled A, C, T, and G. A gene is a special stretch of DNA, a sequence of A's, C's, T's, and G's that codes for something. A gene contains information for a cell to read. But what exactly does that information do? Genes make proteins. Those proteins then interact with each other and all sorts of other chemicals inside the body to build things like freckles, eye pigments, and mood-altering hormones. A single strand of DNA contains thousands of genes or unique protein recipes. Humans have roughly 20,000 altogether. Some genes are small, only about 300 letters long. Others are well over a million. The length and sequence of a gene determine the size and shape of the protein it builds. They determine the function that protein will have inside the body.
Causes of genetic disorders. A genetic disorder is a disease caused in a whole or in a part by a change in the DNA sequence away from the normal sequence. When genes are altered so that the encoded proteins are unable to carry out their normal functions, genetic disorder can result. There's some reason or causes why genetic disorder happen. Genetic disorder can be caused by mutation in one gene. It is called monogenic disorder. By mutations in multiple genes, it is called multifactorial inheritance disorder. By a combination of gene, mutations, and environmental factors, or by damage to chromosomes, it is called changes in the number or structure of entire chromosomes, the structures that carry genes. We also have this reason why genetic disorder happens. We inherit from parents. Most of us do not suffer any harmful effects from our defective genes because we carry two copies of nearly all genes, one derived from our mother and the other from our father. The only exception to this rule are the genes found on the male sex chromosomes. Males have one X and one Y chromosomes, the former from the mother and the latter from the father. So each cell has only one copy of the genes and these chromosomes.
Gene therapy. What if the part of the gene is defective or missing? Or if a gene changes or mutates? It can cause a disease like cancer. Science can replace bad genes with one that is healthy. Add a new gene to perform the function of defective or missing gene. Scientists use vectors or vehicle to deliver a gene. A healthy gene hitches a ride in a vector, and the example of vector is virus. Inheritance. We have two copies of nearly all genes. One from our mother and the other one from our father. The majority of us are unaffected by our defective genes. Only the genes present on the male chromosomes are exempt from its restriction. Males have one X chromosome from their mother and one Y chromosome from their father. Therefore, each cell only has one copy of the gene on these chromosomes. In the large majority of situations, one normal gene is sufficient to prevent all illness symptoms. If a potentially damaging gene is recessive, the normal counterpart will perform all the responsibilities that are allocated to both. Only if we get two genes from our parents, only if we inheritance, we have two copies of nearly all genes, one from our mother and the other one from our father. The majority of us are unaffected by our defective genes.
Only the genes present on the male chromosomes are exempt from its restriction. Males have one X chromosome from their mother and one Y chromosome from their father. Therefore, each cell only has one copy of the gene on these chromosomes. In the large majority of situations, one normal gene is sufficient to prevent all illness symptoms. If a potentially damaging gene is recessive, the normal counterpart will perform all of the responsibilities that are allocated to both. Only if we get two genes from our parents, only if we inherit two copies of the same recessive gene from our parents, will we get a disease. If the gene, on the other hand, is dominant, it can cause disease, even if its counterpart is normal. Only the children of a parent with the condition are likely to be infected, or even then, only half of the children are likely to be affected.
How does experimental gene therapy works? With gene therapy, doctors deliver a healthy copy of a gene to cells inside the body. This healthy gene may replace a damaged, mutated gene, inactivate a mutated gene, or introduce an entirely new gene. Carriers called vector. Transport these healthy genes into cells so that gene therapy has a potential to save 1 million of lives. In gene therapy, doctors are basically hacking the DNA of a living human using genetically engineered retroviruses called vectors. Scientists infect human cells. The retroviruses can be programmed to carry a gene or a little bit of DNA that will override a messed up mutation and make it work properly. The Journal of Science describes one of the recent successes that gene therapy says was really exciting. This is the technique that uses gene to treat or prevent diseases. The most common form of gene therapy involves inserting a new normal gene to replace an abnormal gene. Other approaches include swapping an abnormal gene for a new normal one.
What genes therapy can achieve? Replacing mutated genes. Some cells become diseased because certain genes work incorrect or no longer work at all. Replacing the defective genes may help treat certain diseases. Fixing mutated genes. Mutated genes that cause disease could be turned off so that they no longer promote disease. Or healthy genes that help prevent disease could be turned on so that could inhabit the diseases, making disease cells more evident to the immune system. In some cases, your immune system doesn't attack disease cells because it doesn't recognize them as intruder. The majority are tried. Gene therapy clinical trial were performed in 36 countries from the 5 continents. 4% of this trial were performed in more than one country at the same time. The geographical distribution of trial had slightly changed from 2012 to 2015. American part had slightly increased to 60% in 2012, whereas the European part had slightly decreased to 24% instead of 28.3% in 2012.
So this is the uses of gene therapy. It's a technique that modifies a person's genes to treat or cure disease. Gene therapy can work by several mechanisms, replacing a disease-causing gene with a healthy copy, inactivating a disease-causing gene that is not functioning properly. Gene therapy replaces a faulty gene or adds a new gene in an attempt to cure disease or improve your body's ability to fight disease. So this is the example of uses gene therapy. Clinical genes transfer applications. There are several major obstacles that currently wider, safer, and more effective clinical gene transfer applications. By far, the most important thing is the still nascent nature of the underlying science and technologies, and our incomplete understanding of the mechanism of disease pathogenesis. Genetically modified organism (GMO) can be defined as an organism, plants, animals, or microorganism, in which the genetic material DNA has been altered in a way that does not occur naturally by mating and or natural recombination. The enhancement of plant disease resistance using CRISPR technology. Genome editing technologies have progressed rapidly and become one of the most important genetic tools in the implementation of pathogen resistance in plants. Treatment of cancer and AIDS. The main treatment for HIV/AIDS related NHL are chemotherapy, targeted therapy, and radiation therapy. Chemotherapy is the most common treatment for NHL, was given at lower doses because of the person's weak immune system. Production of transgenic animals. The three principal methods used for the creation of transgenic animals are DNA, microinjection, stem cell, gene transfer, and retrovirus gene transfer. Vaccine development. Each vaccine under development must first undergo screenings and evaluation to determine which antigen should be used to invoke an immune response. Replacing missing of defective genes. In gene therapy, scientists can do one of several things depending on the problem that is present. They can replace a gene that causes a medical problem with one of the dozen odd genes to help the body to fight or treat disease, or turn off genes that are causing problems. Other that future of gene therapy. That said, evidence to date indicates gene therapy has the potential to increase or restore function in affected tissues or cells over a long period of time, and may enable a patient to manage his or her disease without the need for ongoing treatments. Eventually, gene therapy will become a staple of 21st century medicine, but some experts say society will be better served if medical researchers proceed more slowly and prudently.
Genes are medicine. Gene therapy is a new generation of medicine where a functioning gene is delivered to a targeted tissue in the body to produce a missing or non-functioning protein. By using genes as medicine, the underlying cause of disease can be targeted at the cellular level, potentially with just one treatment. Gene therapy is the next generation of medicine targeting the underlying cause of a genetic disease. It has the potential to offer patients a transformational clinical benefits and dramatically improve quality of life. Gene therapy could enable patients to live without the need for ongoing treatments or the burden of daily disease management. Gene therapy is promising, but for now it's only used to treat a few diseases that have no effective treatments. Many types of gene therapy are still being studied to use how safe they are and if they can work to treat or prevent diseases. Goal of gene therapy. Gene therapy replaces a faculty gene or adds a new gene in an attempt to cure disease or improve your body's ability to fight disease. Gene therapy holds the promise for treating a wide range of diseases such as cancer, cystic fibrosis, heart disease, diabetes, hemophilia, and AIDS. The goal of gene therapy is to treat diseases at the genetic level. Gene therapy is a treatment method that is being studied for a number of diseases, including inherited diseases and cancers. For over 100 years, scientists have studied the human bodies and the building blocks of DNA. With each marked progress comes a better understanding of how our bodies work at the genetic level and how we may be able to keep them healthy and free from disease. There are two major types of gene therapy: the gene addition and the gene editing. Gene addition. the addition of genetic materials into the cell to enable the body to produce a functional protein that it could not adequately make before. Gene editing, the process of directly changing or editing a specific site in genome. The techniques in this therapy include gene correction, gene insertion, gene inactivation, and gene disruption. Goal of gene therapy is to treat people with a given disease or condition. Patient safety is a priority.
Now we will talk about delivering desired genes. So what does delivering desired genes means? Gene delivery is the process of introducing foreign genetic material such as DNA or RNA into host cells. Gene delivery must reach the genome of the host cell to induce gene expression. Successful gene delivery requires the foreign gene delivery to remain stable within the host cell and can either integrate into the genome or replicate independently of it. This requires foreign DNA to be synthesized as part of a vector, which is designed to enter the desired host cell and deliver the transgene to that cell's genome. And vectors utilized as the method for gene delivery can be divided into two categories, recombinant viruses and synthetic vectors, or viral and non-viral. When we say DNA, this is the deoxyribonucleic acid, the hereditary material in humans and almost all other organisms. Nearly every cell in a person's body has the same DNA. And when you say RNA, ribonucleic acid carries genetic information that is translated by ribosomes into various proteins necessary for cellular processes. And a host cell is a living cell that serves as a shelter and a food source to the foreign organisms. And the last one is the genome, is the complete set of genetic information in an organism.
Gene therapy is a technique for correcting defective genes responsible for disease development. Researchers may use one of several approaches for correcting fault genes: a serum containing DNA from the damaged gene, chemical treatments to remove the offending genes from cell, and genetics' own unique method for detecting defects in the gene itself that would otherwise prevent disease. Gene therapy replaces a fault gene or adds a new gene in an attempt to cure disease or improve your body's ability to fight disease. Gene therapy holds promise for treating a wide range of disease such as cancer, cystic fibrosis, heart disease, diabetes, hemophilia, and AIDS. In addition, the first approved gene therapy was performed on September 14, 1990 by French Anderson and his colleagues, and it all happened at the U.S. National Institute of Health. It was tested to 4-year-old boy named Ashanti De Silva that was born with severe combined.
What did they do safely? The delivery of therapeutic genes by gamma-retro or lentiviral vector system has proven to be a viable therapy option for a variety of genetic illnesses. In recent years, the number of approved phase 1 and 2 clinical studies is continuously increasing, especially for uncommon diseases. Although the overall obstacles to becoming a widely accepted medication remain significant, clinicians and fundamental scientists' efforts have greatly enhanced our understanding of gene therapies, viability, and biosafety. Despite the abundance of clinical and clinical safety data, we currently lack sufficiently robust assays to predict the exact amounts of toxicity that may be observed in any given clinical gene therapy. When using integrating technologies, insertional Mutagenesis is a big concern.
Here are some ethical issues when it regards to stem cells or gene therapy. The newly introduced viruses may be perceived as intruders by your immune system, prompting an attack. Inflammation and, in the worst case scenario, organ failure may result. The incorrect cells were targeted. Because viruses can infect a variety of cells, it's feasible that the modified viruses will infect more than just the mutant cells. Healthy cells may be damaged as a result of this, leading to additional illnesses or diseases like cancer. The virus is the source of infection. It's possible that the viruses will regain their original ability to cause diseases. This kind of process is called the escape hypothesis, also called the progressive hypothesis. Viruses may have come from broken pieces of genetic material inside early cells. These pieces were able to escape the original organism and infect another cell. In this way, they evolved into viruses. And the last one is the possibility of tumor development. If the additional genes are placed incorrectly in your DNA, tumors may develop.
The four-year-old that changed history in 1989. The parents of four-year-old Ashanti lives with the horror that their beloved daughter was suffering from an incurable gene-based immune deficiency. She had been diagnosed at 2 years old after suffering a string of debilitating infections. This type of SCID would sentence her to a short life isolation, suffering and death. But in 1990, Ashanti's parents, Raj and Vanda Silva, connected with a geneticist, French Anderson, who was lobbying for permission to move forward with the human gene therapy trials.